Herr Snijder, wir haben uns noch nie gesehen. Freut mich, Sie kennenzulernen. Jetzt sehe ich endlich ein Bild von Ihnen. Vielleicht darf ich Sie fragen zu der initialen Testung. Was verwenden Sie da für eine Plattform? Ist das die Plattform, die auch der Giulio Superti-Furga entwickelt hat? Oder in was unterscheidet die sich? Oder ist das so eine eigene Plattform, die in Zürich etabliert wurde? Vielleicht können Sie uns da kurz schon mal vorab eine Idee geben.
Yes, so I was in the group of Giulio Superti-Furga, and I actually was very much sorry. So I'll talk in English, but I understand German.
Yeah, that's fine.
So it's exactly the platform that I initially established in the lab of Giulio Superti-Furga at the Center for Molecular Medicine in Vienna, and I have since moved to Zurich, or back to Zurich, where I initially did my PhD, to become an assistant professor here. So it would be a pleasure to show you a little bit on the platform, some clinical response data we have from a trial.
Is it exactly the same platform you use in Zurich?
It is. So we are actively developing it, but the foundation is identical, yes.
Okay. And how many patients did you screen already in Zurich?
Probably around 600.
Oh, what?
Different kinds. So we've expanded the technology. Initially in Vienna, we were only doing hematological indications, so leukemia, lymphoma, myelomas. In Zurich now we've adapted the technology also for solid tumors. So we're doing melanoma, ovarian cancer, colorectal cancer, brain cancer, so glioblastomas. So we're basically just seeing where the technology can be helpful and where we can actually get it to work.
Yeah. Okay, I think that's the perfect jumpstart for you. By showing this case where the technology has been applied, and we are really very much looking forward to your talk, and are curious what you are telling us on the use and potential of using this platform for clinical decision making. Thank you.
Yeah, thank you, and thanks for the invitation. It's a pleasure to be here. I wish I was there in person, like previous speakers, to meet you in person, but so be it. So as mentioned, I'm an assistant professor at the Institute of Biology of Molecular Systems Pathology at the ETH Zurich, and we're further developing the platform that I previously established in Vienna in Austria, which all relates to image-based drug screening in patient biopsies, on the one hand for personalized treatment discovery, but also, of course, for sort of systems biology and systems medicine. And what you see here is really our daily bread and butter. So these are all automated microscopy pictures of patient biopsies, in this case, a lung cancer of a pleural effusion patient, bone marrow biopsy of a multiple myeloma patient. And here we see healthy donor peripheral blood cells that have all been ex vivo treated with various agents in a high throughput screening manner and then stained with immunofluorescence. And we analyze the data to see what might work. So we call this technology, and this name comes out of Vienna, right? So it has an Austrian history, pharmacoscopy as a shorthand for personal image-based drug screening. The process is actually, the experimental process is fairly simple. So it's really, by now, any kind of tissue that comes out of a human donor or a patient. If it's a solid tissue, we dissociate the cells. If it's a liquid or fluid biopsy, we just purify out the nucleated cells, distribute these in these kind of 384-well plates that has many small chambers, where each chamber can contain a small individual drug treatment or a combination treatment to be tested. And then typically we have technical replicates and different concentrations. We incubate these cells in standard incubation chambers, and within 48 or 24 hours, I lost my slides. Within 48 hours or so, we then fix these cells, stain them with immunofluorescence markers, also based on the pathology information of the individual patients, to identify which of the cells here are putative malignant cancer cells and which are healthy cells, such as healthy immune cells that might be in the sample. We have 2 publications still from Vienna that describe the technology further, but I'll just introduce it here. One of the key differences that we do different compared to previously established drug sensitivity platforms is that we take the healthy cells from the same biopsy as the internal control. So the strategy that we try to optimize is we want to find agents, drugs, combination treatments, immunotherapies that specifically kill the cancer cells, the marker positive cells, while leaving the healthy cells alive in that same image, in the same biopsy. And we show and we know that this works well in one part from classical observational retrospective studies. There's a very small one shown here from bone marrow biopsies of newly diagnosed AML patients, 10 of whom which had received 357 first-line therapy and responded with a complete remission, and 10 of whom which did not respond. And what we do is we expose these cells ex vivo to different agents, in this case plotted again as a function of daunorubicin concentration. And what we score is how the percentage of cancer cells in the uncontrolled condition changes as a function to the ex vivo treatment. So here is plotted with increasing daunorubicin concentration. We see that for those patients that achieve the complete remission, AML patients that achieve the complete remission, we see an on-target cell death of the cancer cells dying specifically in response to daunorubicin. Whereas for those patients that did not respond at the same concentration and time point, we see actually that the healthy cells were dying and the blasts of these patients were surviving. And we can show in a sort of cross-validation accuracy assay that across these 20 patients, we achieved around 90% accuracy to predict which patients benefited from their treatment and which did not. A few words on an interventional trial for aggressive hematological malignancies. This is about to be presented at ASH this year. The abstract can be found online in Blood, and the full manuscript has just been submitted. We also published an interim analysis of this interventional trial in Lancet Hematology in 2017. So what happens here is very much like in the case study that was already just presented. Is we receive small biopsies from individual patients, mostly peripheral blood, lymph node material, pleural effusions, ascites or bone marrow biopsies. Purify out just a Feigl density gradient, purify nucleated cells, distribute them in the 3D forward plates, perform the staining and imaging data analysis, and then return to the tumor board a prioritization of which drugs we see having on-target effects and which drugs don't do anything or perhaps potentially even having off-target effects. And then I just want to show two case studies from this interventional trial. This was an elderly man with an aggressive diffuse large B-cell lymphoma that had relapsed or not responded. Did we perhaps lose the presentation? Yeah. That by the time we received a biopsy, a lymph node biopsy from this patient, the patient had undergone multiple rounds of chemotherapy and also targeted immunotherapy. We tested 104 treatment options based on a lymph node biopsy from this patient and found only a handful, some 5 or 6 agents to show an on-target effect on this particular patient, which included ibrutinib, so the BTK inhibitor. And the patient actually achieved a long-lasting and rather stable complete remission to that single-agent treatment. Another example is a middle-aged woman with an aggressive B-ALL. She presented with pleural effusion, malignant pleural effusion that we received, and of course, there was a lot of material. We could have screened 10,000 compounds. We tested 266 approved treatment options, not just anti-cancer agents, but also other agents. Out of the top on-target acting drugs, it included the proteasome inhibitor bortezomib, as well as the nucleotide analog 6-mercaptopurine, and the patient responded with a strong partial remission to this combination treatment. In all, and what has just been submitted both or will be presented at ASH by Philip Staber from the Medical University of Vienna, we have now some 56 patients treated based on this technology alone, so not including genetics. And these were all patients that had already received standard of care. They were suffering aggressive hematological malignancies, but the cohort spans leukemias and lymphomas, and also includes quite a few patients with ECOG status of 2 or higher, so patients in a poor state. What we report now is that for 54%, so 30 out of the 56 patients, we see at least a 30% improvement in their progression-free survival when we compare it to the progression-free survival of the same patient to their prior treatment. So we can do 30% better than the response to their prior treatment when we just guide the treatment based on such a functional test. I don't have the slide here, but what you see is that there's actually an enrichment for patients with high ECOG values in the subset of patients for which the test did not work well. And in fact, if we stratify or substratify these responses, the difference is significantly more significant for those patients that had an ECOG value of 0 or 1. Now, coming to the Feldkircher case study, as presented before, this was a 69-year-old patient with a primary plasma cell leukemia, 75% plasma cells quantified in the clinic in Feldkircher. We received 4 9-milliliter peripheral blood tubes in EDTA on the 18th of April. These were samples taken just before the start of the treatment of this patient. By our imaging, we imaged around 70% plasma cells in these peripheral blood samples. And I actually found an old screenshot. I didn't have the time to go back into the original data, but I found an old screenshot of the cells of this patient's sample. So you see a lot of nucleated cells, and actually many of these are lighting up in yellow. In this case, yellow was the CD319 or SLAMF7 plasma cell marker. We stained for 2 plasma cell markers and counter-stained for a T cell and a monocyte marker, CD3 and CD14. And based on this material, we performed 1,536 individual drug tests, actually spanning different drug panels. One of the most striking results was in a immunotherapy drug library that's tailored to one of our multiple myeloma studies, is that actually we saw a strong sensitivity to proteasome inhibitor-containing treatments, including bortezomib, daratumumab, dexamethasone, plus minus lenalidomide. So that is consistent with the suggestion. So blue values here suggest or indicate an on-target reduction of the plasma cells. And these 4 rows, they're actually different definitions of plasma cells. So we can score plasma cells by either being CD138 or CD319 positive. We can consider the counter stains to make sure we're not actually accidentally staining or scoring the drug response of T cells or monocytes. And we use convolutional neural networks or machine learning to identify plasma cells in this context. All those readouts say the same thing. These agents look very promising for this particular sample. And as already presented, the patient received, sorry for the typo, daratumumab PTP. This was the time point in which we did the test, and the patient went on to respond well, at least for up to 2 months to that treatment. Now, we tested additional agents, some of the most interesting of which here I'm showing the results of an AML-focused drug library, which includes some 66 drugs and drug combinations. The most potent out of these actually included HDAC inhibitors, so panobinostat, prasinostat, and vorinostat all seem to be extremely potent and also more potent than the dara-VPD at the same time point and concentrations. We saw BRD and BET inhibitors appearing as highly potent for this particular patient, as well as a few other targeted agents and also small compound chemotherapies. That's where I leave the case study, and I thank you for your attention. I'm happy to take questions.
Thank you, Professor Snijder. A question from my point is, can you study immunotherapy in that assay, or is it only chemotherapeutic agents that you can study?
No, so we do actively study immunotherapies. The case study I showed, it did include daratumumab in combination therapy with targeted and chemotherapies, right? So that's, but daratumumab might not be the most exciting immunotherapy out there. We have, especially in the solid tumor field, we have very interesting data on immunotherapies. And this is, let's say, in certain indications for which immunotherapies are, in fact, first-line treatment, we might have data that suggests that we can actually predict which patients respond to checkpoint inhibitor treatments. Biospecifics, trispecifics, we collaborate with pharma companies to test novel biospecifics and trispecifics, and they're actually very potent in our assay. So much more potent than checkpoint inhibitors on their own. The let's say functional assay results from checkpoint inhibitors are rather mild in comparison to responses we see to biospecifics and trispecifics. Yeah.
Can I ask you something? Thank you very much for this very nice talk. So, I mean, I just wondered already when we took over the patient that the colleague from Feldkirch did this functional screening at the beginning. Because outside a clinical trial, I would say this is very experimental. Even though I love this platform, we are performing, you know, we have sent material to Vienna also in situations where we would say there is no standard of care. When you have a plasma cell leukemia, I would say, okay, this kind of 4-drug combination for initiation of a therapy is somehow a standard. It would also be a standard to go for a high dose therapy. And then probably in the first relapse, I would apply such an experimental setting. And the best setting is, I mean, what you mentioned is an EXALT trial or now the EXALT-2 study from Philip, where you are, I think, also involved in Zürich, which we very much support, where patients which do not have a standard of care therapy or an evidence-based therapy, where we randomize between, you know, a physician's choice, genetic-driven and functional drug screening-driven therapy. Yeah. Because, I mean, in this case, for me, it was difficult because the panobinostat in vitro was very effective. In vivo, my experience with this drug class is quite disappointing in myeloma. Okay. And it was, I mean, but now retrospectively, I would say, it was perhaps not a good idea to not expose this patient to panobinostat because it was even more effective than the 4-drug combination in your screen. I mean, can you comment, and also perhaps Bernd, what was the decision to do this already up front? And then can you comment on in which settings you use this screening in those 600 patients in Zurich?
So perhaps Bernd can comment on the individual patient case, and then I can comment on the larger setting.
The testing was initiated from the family of the patient. So, okay. Yeah.
So this is the son who is a pediatric oncologist.
Yes, from Zurich. Yeah, that's right. Yeah.
Okay.
So we had the intention to rescreen the patient upon relapse, but in practice it didn't work out. So this is always one of these considerations. I would fully agree that although scientifically and perhaps clinically it is interesting to see a patient sensitivity at time of diagnosis, so before treatment. The key strategy, as we also followed in the EXALT-1 and 2 trials, is to retest the patient at that point where clinically there is the flexibility or uncertainty of which way to go next. And that what we see is that the closer the test can be done to the treatment decision, you know, the more informative it is. Definitely, and we have additional manuscripts on this submitted. If we look at larger cohorts, hundreds of patients, we just see that inevitable increase of drug resistance. And we don't, so if you look at patients at diagnosis, they will respond to many more agents than as they proceed in their treatment regimen. And that seems to work sort of across patients, across different diseases. So that inevitably means that if the test is predictive, as we hope it is, there are just fewer and fewer active treatment options for individual patients as they progress in their treatments. Right? And I think that's so even statistically, just the chances of finding something that works becomes smaller and smaller. And we're actively working on the underlying molecular biology to understand what the general resistance mechanisms are.
Are you doing whole exome sequencing in parallel from all these patients?
Yeah, so we have certain studies where we exactly integrate with whole exome sequencing. For instance, colorectal cancer. The absolute challenge in there is that you find a lot of variations and variants that haven't been clinically annotated. So it's whole exome sequencing is just extremely challenging to really link first to what we see. We would actually need something in the order of 10,000 patients of a certain type to start being powerful with our associations there. And but we have studies where in larger collaborative projects, something is called the tumor profiler, which is sponsored by Roche. We actually integrate with pretty much every molecular data type that we can get. So transcriptome, proteome, cytoph, single cell RNA sequencing, DNA sequencing, whole exome sequencing, including FMI from Foundation Medicine, and the functional drug testing that we provide. And that, it provides a wonderful research, but it's a huge challenge to actually on the fly in a clinically relevant time frame digest that data, right, to something that makes for actionable treatment recommendations. And one of the things we see is that the drug testing on its own has a certain kind of simplicity, right? For an NS1 patient, you might not need 100 patients before you can say, did that work? It's ex vivo, this is what we saw being active. So there's a simplicity in the kind of data that we measure for the clinic. And we're seeing if it translates to improved treatment responses. Yeah.
But you're doing, that's really a great job. So I'm fascinated by the technology, but I think we have to integrate it into the well-organized clinical trials. I'm really happy that you are starting and have initiated together with the Viennese colleague the EXALT-2 study, which is very important to show the potency, to really also show the clinical benefit.